Hello! Welcome to Microbiology Journal Club. I'm Faz, this is my colleague Danny Chan, and we're here to talk about some microbiology.
Yeah, we're really happy for everyone to join us. We've been doing this for every week now. We have a system where we have off weeks where we look at the news titles. That was last week. And then we have our on weeks where we dive deeply into one of those papers and do a peer review process where we look figure by figure and try to figure out what are they teaching us.
Yeah. So this week we're going to be looking into Neuropilin-1 facilitates SARS-CoV-2 cell entry and infectivity.
Hmm.
So,
A new
um, yeah. So this is so to flesh out this a little bit, I'm going to move on to like our first slide, which we're doing some background to help explain kind of some of the background to this.
Absolutely. Oh, I mean, I guess on our website we've written like we have like some general ways in which that we approach journals
Yeah.
and knowing the background of right like you have to be able to contextualize what the results that you're looking at within the wider scientific literature. So yeah, our first slides are just to try to help do that. And actually this is something we read previously. Hoffman et al 2020 was a paper that basically use cell culture to figure out that they could inhibit the entry of SARS-CoV-2 with a specific type of protein or a specific type of cell inhibitor. Um, and they figured out, well, I think other people figured it out, but I just picked it out from this particular paper that ACE2 is the receptor for SARS-CoV-2, right? So like
Yeah.
in A, they have like a bunch of different pseudotype particles that they're trying to see if the cells express human ACE2 or bat ACE2 or some other receptors, which ones get in. And then in B, they like show basically a very similar thing. They can block entry by using an anti-ACE2 antibody. And then in C is like, to me, that's like why I chose this whole slide to show, right? Is that they show you that when you add ACE2 into a cell that didn't have ACE2 before, or very low levels of it, you get infection by the actual virus, genome equivalence being a measurement of the actual virus. I mean, that's what we use diagnostically, right,
Yeah.
in that RT-PCR test.
Yeah, I mean, it's kind of accepted that ACE2 is like the main receptor for SARS-CoV-2, and to the point where we've looked at papers where the treatments are based on that idea that if you block ACE2 using something that looks similar to like ACE2 or binds to the SARS receptor in the same way as ACE2, it's going to stop
Absolutely.
SARS infection. Um,
Absolutely. Yeah, in the next slide, like that's exactly what they show. They use this camostat, and we know that actually blocked. It actually blocked proteolytic processing. So right, it's like
yes.
it's not just you. So like I guess in the previous one they use an antibody to block it. Right, that means like you can't bind it because there's something in the way. But there's a whole process to getting the spike protein ready to enter the cell.
Yeah, so spike protein has like two conformations. So when it's binding, it's in one conformation, but that binding allows it to be cleaved by TMPRSS2 in this thing called a furin cleavage site that causes it to split into a fusion peptide. So part of it ends
Yeah.
up fusing into the membrane, and the other part gets cleaved away.
Yeah.
Um,
So yeah, I guess it's like like when you make proteins, right? You make that big noodle,
yeah,
but sometimes for the specific functions, you need other pieces that are exposed, and so this cleavage, right, sort of allows the right peptides to be exposed that then facilitate the fusion. And so
yeah,
in this slide, camostat blocks one of those proteases.
yeah. So it's kind of like almost like the trigger. So like kind of it goes so almost like it's like an elastic band that's holding it together, and then when you close it, it goes springs out and triggers the entire process of this virus membrane and the host membrane coming together and fusing. Uh,
Right. Right.
uh,
And so, yeah, to zoom out a little bit, in the next slide, we're talking about ex vivo human samples. I like this. There's actually not a lot of stuff like about like human cells, right? Like in
yeah,
like realistic conditions, getting infected by SARS-CoV-2. So ex vivo, for those that don't know, it's like the Frankenstein, I guess, of cell culture, right?
you
you
take a look.
Oh yeah, take a
You take
little
a
bit of
the
the actual
flesh.
thing. I think they use autopsy. These were like recent cadavers that they
Right.
used, um, and they place them into cell culture, and then you can do whatever you want with them at that
Mm.
point, right? And they infect them with SARS-CoV-2 as well as classic SARS in this
Yeah.
example, and they stain using like something that makes a red color. So all that red that you're seeing, those are infected cells. And you can see like in the bronchus, like that whole epithelial layer gets red. In the lung, you get all the spottiness. And they actually found that even in conjunctiva,
Mm.
which is the right in your
Yeah.
eyelids area, that kind of moist membrane is
Yeah.
susceptible to SARS-CoV-2 entry here.
Yeah. So that's why like always wash your hands and rubbing your eyes. You don't want to get SARS in your eyes.
Don't do it. Very
Yeah.
important tools that we have in preventing the spread of the virus. Don't let the droplets get in your mouth. Don't let them get in your eyes.
Yeah. So on the
Um,
next
yeah,
slide, we got like this. Can you take me through these diagrams?
yeah, absolutely. In this one, I found a review because there's so much that's been done now
Mm.
in this field, right? Like we've both experimentally and within the population.
Yes.
Um, there are just like so many examples of seeing like, oh, this infection occurred, positive people, right, showing
Mm.
different symptoms, getting biopsies and seeing like where that virus occurred. Also just swabbing every part of your body, right? Human beings who have had SARS-CoV-2 have been swabbed at every orifice and opportunity. And they found out that there's sort of this heat map that shows you as you get darker in tone. The blue, at least, is where they found SARS-CoV-2.
Yeah.
So they've been finding this virus all over the body in these different places, um, and this is called I guess broadly in pathogenesis we call this tissue tropism, right? So the ability to infect or replicate in very specific tissues. They have some affinity towards it, right? Every virus is a different tissue tropism, and
Mm.
that's defined by its molecular outset, but it's also defined by the receptors that it uses. And so in the second diagram here, they have ACE2, and like you get darker, right, with ACE2, and this is where ACE2 is made in the body,
Mm.
right? So very well accepted that ACE2 is the receptor, but when you look at the overlay between where there's the most ACE2 and the most virus found, it's a little bit different.
Yeah. And that's odd.
Yeah.
So yeah. I mean, you'd
So
expect it to map, but
you'd expect it to map if right, and that's like given this big assumption, right, that ACE2 is the only determinant of viral tropism,
mm.
right? But that's not the case, right? Like there could be, well, as we'll learn in this one, there could be other co-receptors, but also
Yep.
like different cells express different things in the body, and
Yeah.
because viruses have to work in basically the system, right? They are hacking us, right?
Yes.
Like
Some
because
areas
viruses have
aren't.
to work. Hmm?
Oh, some areas just aren't accessible to viruses. There has to be like delivery where they go to, and like
Where they
they have
go
to.
to, yeah.
So,
And is that cell like going to make the thing that they need to to
mm.
to enter that process, right? And so um or some cells are also well defended, right? That's another
Yeah.
that's another thing that can drive tropism. Yeah, so anyways, there's many determinants of viral tropism. That's why this picture doesn't match up 100%, right? And I guess that will launch people to wonder, are there other receptors?
Yeah. So if we go to the next slide, we get this.
Yeah.
So I think so. This idea of this C end rule pep motif. So a
Hmm.
couple of years ago, some researchers did a massive protein screen and looked at and found that there's a certain motif that binds to Neuropilin-1 and other kind of proteins, which is essentially like an R gap with an arginine between two other peptides, another arginine at the end
Yeah.
of a peptide. And
Yeah,
one,
so that's the R is arginine in this, right?
yes, R is
P is
arginine.
P is proline, and then you said like XX, right? Like that's how they describe this rule.
Yes,
XX meaning anything, and then at the end there also has to be an arginine R, right? And something. Yeah.
yeah. So I mean, I've thrown up
Oh, an
a
OH.
yeah, an OH. So that's just the end. So this works basically as long as the exposed end of the peptide. So the C terminal and like N terminals, they tend to stick out a little bit from peptides. So things can bind to them in a certain way that they can't to other peptides. Um, the thing that's very interesting about This is the R like gap R is also very similar to the furin cleavage site sequence, which is an RRAR. So the hypothesis
Hmm.
is so once the furin cleavage site is like snapped, can it bind in the same way as the endral sequence?
Yeah, absolutely. Yeah, I find that this is like the most interesting. It's the most interesting way to start the hypothesis, right?
Yeah,
Because like they found this sequence through just a screen of like
yeah,
they're just like show me all the peptides in the world, right? And show me which peptides bind to neuropilin. So that's not even in like the infectious disease field,
yeah.
right? That's like totally in like an abstract biochemical field where you're like I want to know like things that bind receptors. I want to know
Hmm.
what peptides look like, what peptides drive binding. Um, and from that field and this kind of observation of could there be other receptors of things, right? You can build the hypothesis: does the SARS-CoV-2 spike protein have such a have such a have such a binding domain and thus have an expanded receptor repertoire?
Yeah, and so like we've got a diagram of SARS-CoV, the spike protein up here, and you can see the S1, S2 is the furin cleavage site, and that's kind of on the outside a bit. And there is another like cleavage site that isn't talked about as much, which is the S2 prime. We might talk a little bit about that, but probably won't. But that kind of illustrates that there are that. I mean, the question is like, does it even need to be cleaved in order for it to bind neuropilin? So that's something that we.
Right. Sure.
So yeah, moving on to the first figure S1, which
Hmm. Yeah, they're gonna give us just to set up the tools, right? Supplementary is a lot of like tool setting up, right? Tool validating, and then sometimes like analyses that don't fit quite into the story but support it.
so
uh, and in this one, they're looking at cells that express NRP1. So they have two cell types,
hmm,
KCO2 cells and HEC293 T cells. Actually, A and D are good ones to look at, right? Because
yeah.
you can see that CACO2 cells have a lot of ACE2 and a lot of NRP1. HEC293s do not have a lot of
Yep.
ACE2 and not a lot of NRP1.
Yeah, and they
Uh,
use a special version of the CN. So the CPAPR is like this kind of like almost enriched version of it, where it has like two of those motifs. So in previous work, they found that
mm,
that bound like extremely well compared to average CN rule ones. So this is like
yep,
a good demonstration of like that specific binding. So this is almost like your
absolutely.
first step. So like
Yep. And again, this was what they found in previous work when they were just like screening for random peptides that bind, right? So
yeah,
this is a positive control, right? As we go into our papers, right? This is like we know this binds NRP1,
yeah,
and they attach it to this like silver nanoparticle so that they can see what ends up taking in those particles as a proxy for knowing the binding.
yeah. And like so they find HEC 293T is very bad at both of them. So and they also find it's
Mhm.
also kind of bad of SARS-CoV-2 growing in it. Uh,
Yes, absolutely. Yeah, and you can attribute that to the ACE2, right? Like because in F, right, they take those hex cells and they put ACE2, or I think, wait, is that what they do, or do they use blocking antibodies? I think actually that's what it is. Let's look at that figure legend. uh, oh, I thought I had the figures open in front of me.
let's see
Oh,
what the
yes, I do. Supplementary materials, figure one. I should have scrolled down this before we began.
oh it's
Not
it's
to
it's no
scroll through.
problem. So let's see.
Oh wait, F. I got it. They
So
are caco cells
yep,
incubated. So caco cells, they have the ACE2. They're incubated for 30 minutes with PBS or ACE2. I guess that's soluble
yeah,
ACE2 they're
soluble ACE2.
using.
So
So
the
that's
idea,
actually back to the blocking
yeah,
methods that we thought the decoy.
yeah. So
And so the
yeah,
idea is that because the ACE2 just floats around and binds to all the SARS-CoV-2 pseudotype particles, in here they're not using the virus, they're using pseudotypes. We've
mm,
talked about that before. Basically, very similar to the virus, but not the virus exactly. Yeah, it saturates all the spike proteins, and then so the viral particles can no longer bind to their receptor, right? Because they're already
mm,
bound to their receptor, pre-bound to the receptor. And so, anyways, we've seen this result before, right?
yeah,
Yeah, the binding is mediated by ACE2.
yeah. So this kind of demonstrates the first step that, oh yeah, these bind to NRP1, at least, and CN rule works for these. Um,
Mm-hmm. Mm-hmm.
so next slide is the figure 1A. Uh,
Mm-hmm.
so it
Oh, and I
looks at,
guess it's the same thing. They also show it here. I didn't have to
yeah,
explain it in this.
pseudotype SARS-CoV-2 with and VSV-G protein. So that's a different protein that's like they're using to compare. So I
Yeah,
mean, you look at
that's
that,
the positive control. It uses a
yeah,
different receptor to get into the cells. A
yeah.
different type of positive.
So
And
yeah, yeah,
so
the purple cells,
basically, it can get in.
yeah, the purple cells are the positive. I mean, I mean, a lot of these. So the big question I had for these is like, where do the graphs come from? So is it from so because it's not necessarily quite clear in the methods whether it's either they looked at I mean I think they just looked at a bunch of different microscope slides and then took the kind of numbers from those. So I think they took like
Yeah.
a sample of three and then did an average from that.
Hmm.
I mean,
Oh yeah, the graphs themselves. Yeah, I don't know. I mean, definitely three, right? Because there's three dots in
yeah,
those in those in those bars.
yeah.
So yeah, I assume that it's the those are very small experiments, right?
Yeah, I would have liked to see flow cytometry on this because that's another way that you could have done this kind
Flow
of
cytometry is
experiment.
a great way. Yeah, absolutely. But yeah, they're moving
Yeah,
quick because they're gonna build a story. Hopefully, we always ask for other methods, right? Because
yeah,
like one way of showing things that's great. Two ways of showing things, it gives you a lot more confidence that what you saw isn't a fluke.
yeah.
Because even you can run as many statistics as you want on a single method, right?
Hmm.
The chance of it being a fluke is still like if that's from the method itself, you're always going to be stuck with those, right? Those statistics, right? Like it could still just be wrong because of the method. Um, and so, yeah, always good to do different methods. They don't do it,
Yeah.
but
So
that
basically,
would
yeah,
be the
there's
highly rigorous way of approaching it.
yeah. So yeah, I mean, some of the cells are expressing NRP1, and but it's not quite sure whether there's much more than say control or. But yeah, see, I guess so. We'll move on to the next slide
Yeah.
where we've
So,
got
wait, some cells are expressing NRP1. Yeah, like because in this one they're testing each of these proteins by themselves in the
yes,
hex cell, right? Because we know the hex cells don't bind at all. So like, and here they also bring in that TMPR SS2, which I guess we should say like before that's the protease that pre-processes the spike protein.
yeah, that's the thing that hits the furin cleavage site and causes it to go into the other in the fusion conformation. Um,
Yeah.
And yeah, so the thing is, it is already because I think the previous slide in the figure S1 they show that it is expressed in HEC 292 cells. So,
That's right. Yes, you
but it,
have this protein TSS2 is how they're abbreviating it
yeah,
in G. Yeah. It's in
uh,
HEC and CACO cells.
yep. And so we're moving on to Fig 1B, where again we're looking at those purple spots to see which where that shows us the things we want to see, which are the
Yep.
cells that would be uh,
Get infected.
infected. Yeah, yeah.
Yeah,
In
with
fact,
pseudotype. I
yeah,
don't
with
think they ever
pseudotype.
really go into the full virus until a bit later. A
Yeah,
lot of these early biochemical experiments, they're done with pseudotype virus. But
yeah,
I guess we should just briefly say it since they use it so much, right? It is a viral particle, right, from a different virus that
yep,
has the spike protein on its outside, the SARS-CoV-2 spike protein.
yeah.
I actually think that to me that's one of my pet peeves about this paper is
Mm,
that they say SARS-2 right as
mm,
like their experimental condition, but they're talking just about the spike protein.
yeah,
Right? It's not SARS-2. It's a pseudotype particle that has the SARS-2 spike protein. Okay,
yeah,
anyways, just a pet
yeah,
peeve of the way they display it because it means that when we extract these images for people, right, out of the context of the paper, you can mislead people and people will be like, "Oh, they did those experiments with SARS-CoV-2." Hold on, pseudotype particles.
yeah. And I mean, the cool thing is, they show it in the graph that it's not so much that the NRP does ramp up infection by the looks of it. That if you got ACE2
Yes.
and TS2 are good enough to establish an infection, but NRP1 increases it.
Oh yeah, yeah, in this one.
Yeah, like a value added virus
Yeah.
receptor.
They added a lot.
Yeah.
Okay, moving on to
Let's
1C.
see.
So here we're looking at NRP1 on those CACO2 cells, and we're giving them infections. We're
Yeah, well,
saying that we know CACO2 cells already support, right, a high level of infection, right, uh, it, compared to the hex cells, and we're saying if we add more NRP1, we get
mm,
even more, even more of this.
yeah. That I think
Yeah.
that's basically and 10 basically that illustrates the model. So this is from Daly et al., which is another paper that's published at the same time on basically the same subject, and we kind of tossed a coin and chose this paper instead. But
Yeah. Well,
they have
I remember saying that like we like that this one said cell entry and infectivity because that's like a more like definite phenotype that we can criticize versus
yeah,
host factor is kind of like a nebulous thing.
yeah, host factor. Anything can be a host factor. This actually showed you what it did. It clickbaited me in basically.
Yeah.
But yeah, they delve in a bit more about the binding of the CNR to the Neuropilin-1, and they
Hmm.
kind of illustrate this model, what's happening a bit more clearly. What they think is that because I mean, they think that like essentially like the S1 like the cleavage site gets cleaved and doesn't all like float away at the same time. Because it's in a trimer. So
Yep.
so when one part's cleaved, then that can still bind to a neuropilin, and then
Hmm.
that can help facilitate infection.
Yeah, I like this. I like it because it shows us the difference between the cleavage site and the receptor binding site,
Mm.
right? Receptor binding site, like that exists somewhere else, but it also shows that like you do need like having we know right that having these cleavage proteins furin or TPSS right in close proximity is going to help this process go forward, and they
Mm.
kind of give you that spatial awareness, right, that like oh it's because like that means that it's then closer to bind something else.
Yeah.
One thing I want to say about this model, because I've been noticing it, right? Or I notice people, basically my own understanding. When I've been thinking about furin and TPRSS or whatever, I think about it as happening outside of the cell, right? Because
Mm.
like those are proteases that are outside of the cell, right? That are acting on these spike proteins, right? But
Mm.
during like endosomal, when the endosome forms, right, and grabs a portion of outside of the cell, you can still like do enzymatic reactions in that zone, right? So it doesn't have to be strictly outside of the cell.
Mm.
Outside of the cell meaning outside of the cell on the membrane side, but you could be in an endosome when all of this happens.
Yeah.
And I do believe we did see data like that in the previous Hoffman paper because they use like an inhibitor that was known to inhibit endosomal proteases.
Mm.
And so like right like this process, yes, it's happening on the outside of the cell, things are washing over, but it's also like happening like as the cells grab as they bring in parts of the outside, right? It could be happening there on that interface. I just wanted to say this
Yeah.
picture reminded me of that when I saw it.
Uh, yeah. I mean, we get that a lot with the way we represent science. Is that we try to make things simplified, and sometimes that can make things almost too simplified. So we get this kind of weird idea of how things are.
Yeah, absolutely. And I think like because I want it's nice to have that model to know that it's not just like on the surface of everything, right? That some things could also happen on the inside. It's just they're not totally in the cytoplasm yet.
Yeah.
Okay,
So
so
moving.
yeah, moving on, S2AB. They're going to show us some tools that they can use to study this interaction. They have an antibody, basically, that blocks the binding between the domain of NRP1,
Yeah.
which they call B1, B2,
Yep.
uh, and the peptide, the the nanoparticle, the nanoparticle
Yep.
coated. Yeah.
See par par par.
See, and that just reminds me of the pineapple pen thing. But yeah,
uh,
so they show us the antibody does block, right? When they use it, they get less fluorescence. They show us an image of the same thing. That yeah, it's working. Let's move on.
yep. Move on. And figure S2C. They checked out different antibodies. So they had three different monoclonals that block this binding area, and they found that their third antibody, MAB3, is the best. Uh,
Yep.
And these are
Yeah, just
like
some actual assay pictures of it, right? They showed us bar graphs before, but now they show us
yeah,
pictures. This one is binding to the outside. I guess it's just how long they waited,
yeah.
right?
So yeah, they
Yeah, they if they take the image early, then you can see it coats on the outside. If you go to the next one, you can see that you wait a bit longer and it goes to the inside.
yeah. I mean that's an important thing to also notice that like it does decrease the internalization a lot, but it's not completely gone. So that's a thing to consider when you're looking at the other data. Just
Yeah,
um,
antibodies aren't perfect, right? Like they're gonna
yeah,
have some background still that this works on. And then they give us a picture, E, basically just showing us the same thing. Yeah, quantifying it for us.
yeah.
How is E different than? Oh, a different cell type probably.
Yeah, because they do use PPC for the
Hmm.
big like images, and the small one they use like I can't remember which one that they use, but you can check
Probably
quickly.
Caco. I think it's
Caco,
Caco cells.
yes, good old Caco cells. Um, so
Cool.
cool. Yeah, we can move on to the next slide, which is Figure 1D, where they take this MAB3 and they look at see whether it reduces infection by the pseudo
Yep.
particles.
By the pseudotype particles,
Yep.
and sure it does.
Yep.
uh,
Yeah, basically, I think about as a right-hand images are like the baseline infection, and then the antibody kind of brings the cells closer back to that baseline infection by blocking that kind of binding.
Absolutely. But what I love is this F, right? Because we're getting now to a bit of the meat of their observations. Yeah, it's because that effective decrease is only present, right, in the presence of NRP1.
Yeah, exactly. Um,
Yeah, but you can see the scales are different, right? Like because
hmm,
we know that the NRP1 added to the cells boosts the infectivity tons, right? So
hmm,
indeed that scale's huge, right?
yeah. So
Like
yeah,
there's just a lot more infectivity. So you remove that and you get less infectivity, but maybe there's still an effect, right? If the antibody is
yeah,
working, but it doesn't, right? Because the antibody specifically blocks NRP, and yeah, so you don't see that decrease. I like it.
yeah. I mean, as we've seen before, the antibody isn't necessarily perfect. They can't do everything. Stuff still leaks through, but it kind of established that trend is there. I think.
Hmm. Hmm. Hmm.
I anyway.
It's the necessity of NRP1, right? It's like
Mm,
they basically show it two ways. It's like the antibody blocks, right? But the
mhm,
antibody only blocks a certain thing.
right.
And
Next
then
slide.
here,
Sorry.
yeah, next slide. So I guess they're bringing us to back to the particles now. The right?
Yep.
Or oh no, they do pseudotype first. And they're using the soluble. They're doing another type of blocking experiment, right, with the soluble domains of the NRP1. So just like
Yeah.
how you saw earlier, you can block ACE2, right, by just giving a bunch of this soluble protein. They give the soluble version of their NRP, basically NRP binding domain.
Right.
And I love it that they have the mutant of the wild type. You know, they already had all
Yeah.
this stuff in their toolbox, so they're just showing us another way, right? That you need the wild type binding ability of NRP1 to stop the particles from being able to bind normally.
Yeah. This is like the best part of the paper where you realize the moment you say no shit Sherlock is a point where they've just convinced you of it. You're like,
Yep.
why these experiment? They've already proved it. That's where they've passed your threshold of skepticism. And I feel this is
Yeah.
where they've done that for me, where they've done this extra experiment just to, and then like I'm like, oh wait, the reason I did this is because there's all these other things that if I was a proper skeptic, I'd be able to pick up and go, do they need to do
Yeah,
that? So
absolutely, absolutely. It's about being rigorous in science, right? And it goes
yeah,
back to this thing I said before, like you can show me once and the statistics could be amazing, right? But it
yeah,
doesn't matter unless you show me in another way. Because like statistics are statistics, right? There's still a small percent chance that it's wrong, and if you can't address that through an alternate means, then it doesn't make any sense. Yeah.
yeah. I mean, because there is a concern like with these mutant binding domains where they've altered like three amino acids. So
Hmm.
the antibody doesn't bind to them, but does that mean that they still aren't functional? Can they still bind the CN rule domain? And I think this
Hmm.
much more clearly demonstrates that.
Absolutely. Yeah, it gets at a nuance of the binding, right? We're showing
Yeah.
because through the methods of showing all these different bindings in different ways, there's nuance that we're gaining. Yeah. Okay, so let's launch into the next set of experiments.
Yep. So
So they have an uncleavable spike protein, is what
mm,
they've developed here as a tool to launch us into the next set of analysis. And the way they do that is they passage their actually it's now SARS-CoV-2 itself, right?
yeah.
They're passaging an isolate of SARS-CoV-2 in that they could grow in caco cells, but they passage them in vero cells until they lose the ability to be cleaved by furin.
Yeah, this is an interesting because we did touch on this in a previous article we looked at where they looked at all these different cell types and found that vero cells could always get like infected by SARS-CoV-2 no matter like what the situation, and that means and
Wait,
I've looked
was that
and
the insect bat cell thing?
yeah, I think it was the insect bat cell thing. So I think
Oh
that
my goodness! Yeah, that's a
yeah, that's
that's
one of our
crazy.
first like videos was on this whole thing,
Yeah,
and the reason why
and
that's
that's interesting
important.
too because that has really no bearing on right. That has so little bearing on the therapeutic side of things and like the yeah. But it does show us yeah. I guess because it's like biology, right? Like we're going through different ways of learning the basic biology. It becomes relevant when we're discovering things like this, new biological interactions.
Yeah, and I think it's an important consideration for because a lot of these like vaccine challenge papers, a lot of Tropism, they grow the SARS-CoV-2 in vero cells. So in so many things I've seen, and
Hmm.
like this is a real problem because I think the paper that discovered that this is an issue is like made the point that we really need to keep an eye on this because if you passage them too much, then they don't represent anything like in reality. So you could end up seeing papers that end up being a lot treatments looking a lot more effective than they actually are because you've
For
evolved the
sure,
virus to live in a
for
cell
sure,
culture rather than
for sure.
in reality.
Or yeah, I mean, I think the other obvious thing is that we can't when you see cellular assays and they're done in vero cells, right? That is not very realistic for a human cell condition, right? Because we know human cells have all this other stuff, right? And
Yeah,
obviously, like in this paper, we're sort of quantifying some of those and describing it for people.
yeah, because it's I mean furin cleavage is not an obstacle for infection in vero cells, so they could be going infected in some other way, and because of that, it means that uh, SARS-CoV-2 that doesn't get cleaved in the furin that actually ends up predominating. So you get
Yeah.
these uncleavable spike proteins
Well, actually, I
evolving.
think if we go to the next slide, S3, you get that image from B, right? Like here's where they then take that population that was unable to cleave, right? Because like not all the viruses changed in a single
Mm,
passage, right? But from after all six passages, that viral isolate, right? Um, they could figure out that a certain percentage has the mutant, a certain percentage has the wild type spike, and when you put them back into CACO2, they actually go back. You end up
yeah,
selecting for the wild type spike, meaning that the wild type spike has an advantage, right? In this condition, evolutionary advantage. I mean, what is it? I mean, of course, they're bringing us to the suggestion that it's that NRP, right? That additional binding that it gets by binding the NRP is giving it that advantage in CACO2 cells. And A is like some weird sidebar, I guess. They're showing that it's furin cleavage
yeah. So
that yeah that is being selected against here. In some ways, I think S3A is the reason why we get S2 or 2A, right?
yeah.
Is supporting 2A. All
So
right,
now
yeah. So let's.
22ST. Sorry. So what happens if we substitute NRP1 and ACE2? Um, So it seems like both of them have some uptake, but NLP1 is like less effective. You need many more viruses to boost the chance of NLP1 cells that are expressing that alone in order for it to
Yep.
treat. But the fact that it can do it without ACE2 present is really interesting.
Yeah, yeah, yeah. They can do it without ACE2 present. We saw it already earlier, but in this one, I guess the difference is that they're showing us that as you add more virus, right, you see more of this effect. So the effect is there. It's just kind of low, right? Of
Right.
of basically NRP only entry.
Yeah. So you really, so this is, it's not going to be the initial cells you get. This is going to be like once you have a full blown infection, you have enough viruses around there, you get a situation like this where
Sure.
potentially, I
Yeah,
mean,
yeah. I mean, that's the implication that they're trying to draw for us, right? Is like this could be happening during disease. Okay. But I guess the other thing is that I don't know. I guess this is also a test of their stably transfected cells because I
yeah,
guess they never were working with the cell line before. Yes, they were working with HEC 293 cells that were expressing these different proteins, but it wasn't stable transfection.
mm,
And this is something we can speak to like as experimentalists, right? Like
yeah,
at the beginning, they did all these experiments with just transient transfection because they didn't know what was going on. They were
mm,
just testing hypotheses. But when they see the inkling of something being like, oh wow, like this could be an interaction that is meaningful, well now you're gonna have to do tons of experiments with the cells that express that protein. And so you don't want to transiently do that anymore. You want to make a
yeah,
tool to help you do that.
yeah. So 2EF we're going to, so,
Yep.
uh,
2EF. Yeah, so here now they're going to use those stably transfected cells, and they have sort of a triple, right, with everything together: NRP, ACE2, TSS, TMPRS, and then they have one version that just has the TS and the ACE2, and they're showing us again, right, that there's that additive effect of having
mm,
NRP1 in the system.
yeah. I don't really have anything to add to that. I mean, um,
Yes. Oh no, but no. I see, but now they're bringing in that tool, right? That the cleavage deficient mutant tool,
yes, yeah,
right? And they're
and
saying that in the cleavage deficient mutant, right? You don't get that increase when you add in the NRP1,
yeah, and you
right?
get a
The great
slight increase with
numbers.
TSS too. But
Hmm.
I think I did want to make the point because I did highlight earlier that there is an S2 prime site that can also get cleaved, but maybe
Hmm.
less often than so by other cell proteases. So there can be some background cleavage going on as well. I
Sure.
haven't really found many papers on that S2 prime site at the moment, but I'll dig in to see what else I can find maybe. But yeah,
I mean, again, like I guess what I wanted, like, because this figure, it's figure two now, right? We've moved from figure one, but it's like beating the horse, right? Like it's showing us all the different ways, right, that they can show us, right, that this interaction is between some peptide, right, and NRP1,
yeah, um, and
and that
figure.
peptide is from the cleavage, right, and that peptide is
Hmm,
from this cleavage site. Sorry, I just I'm trying to like say like, am I
yeah.
covering exact statement of these figures? All right.
So uh, all
Oh,
right.
same thing.
Yeah, same thing
Right?
basically.
Now CACO2 cells, right? So now they have their one where they put the proteins in. We know CACO2 cells already have NRP1, ACE2, and whatever, right? So can we see the same thing that the effect is only really seen with the mutant cells, right? The infectivity, and here it's sort of the opposite. If the mutant cells are unable to, right, uh, be affected, then yeah, it would tell us that again, it's the ability to cleave this site that is generating something that binds to NRP1.
Yeah, I mean, this is an interesting one because I mean, looking at the cell images, it's quite difficult to actually spot differences because again, because
Hmm.
you do need a bit more of a. I mean, it's hard to present this sort of data in one paper because you'd really want to give like a massive image of everything or have something like a.
Yeah,
So
absolutely. Well, I mean, it shows you that like this is where like this is why like as you just can't show images, right? Scientists can see things, you have to be able to quantify them
yeah,
afterwards. Hmm.
um, so
Yeah,
figure.
and then they use the right. All right. Yeah, sorry. uh, so they use the antibody here, right, to block that interaction. Yeah. So antibody doesn't block the or
The
antibody blocks the wild type
but
infection, right? But not this weird cleavage version, right? Because gray
yeah,
is the, the unable to cleave.
yeah.
Hmm.
So it kind of highlights again that how important the cleavage site is to that level, the role of certain proteins in infection.
Hmm.
Um,
Moving on.
moving on. Yeah, so we've seen silver nanoparticles with the like perfect C-end rule peptide, but now they've got ones with the C-end rule peptides that are on the SARS-CoV-2. So they actually like use the different variants that can occur when the spike protein is cleaved. So uh, they list them like, so there's one which is just like how it should look after furin cleavage, one that looks like how it should look before furin cleavage, just in case that this binding is happening to the whole to that, just to check whether the binding can still occur when you've got no cleavage happening, or if it's been cleaved at a different site. Uh,
Yeah, yeah, different. They're basically testing. Yeah, they want to see the cleavage in action,
mm,
right? Or like, oh, I see. There's different potential cleavage ends is what they're looking at here.
yep. So they've got like one with an amide group. That's with an
But they
H2 group.
but they made them all synthetically,
Yeah.
right? This is not actually from a cleavage event. They're just
Mm,
sort of running through variations of a cleavage event from the actual cleavage site of SARS-CoV-2,
yeah. And
which
they're
we
doing.
know conforms to the C-end rule.
Yeah.
Yeah.
Yeah. We've been convinced of that by now, and it kind of shows that the only authentic like post after furin cleavage sequence seems to be the one that really works.
Right, right. Okay, so moving on. Yeah, so all of that to say, right? Because I guess to me, like up to this point, they were working from an abstract idea, right, of the
Mm,
prototypical C end rule motif, and they needed to connect that to like, the actual motif that is found in SARS-CoV-2.
yeah.
And
So
through their experiments, they've gotten us to that point.
yeah, this
you
is the point where
I would agree.
we,
Right? Yeah,
yeah,
I'm convinced.
I'm convinced it can happen in the petri dish in those cell cycles, but would it work in a mouse? Would it work in a real situation?
Of
In the
course, always, because we've only been given these models, right?
yeah. So yeah, I mean there is like a thing that always like happened in work in progress. Like whenever you're talking to the supervisors, like, oh, but would it work in a mouse? Would it
Yeah.
work in a human?
Hmm,
Because again, there's always something unexpected that turns up. So we can build up
absolutely.
these brilliant models, but then once the rubber meets the road, suddenly things can happen. So it's
Yeah.
great that this is where the researchers are taking it to the next level, where they are now looking. This is just figure S4 is where they're looking at the NRP1 distribution across the mouse olfactory epithelium and in the olfactory bulb. So
Hmm,
epithelium is like the surface of the nose, and olfactory bulb is like the part of the brain that handles information from the nose.
yep.
And in mice, it takes up quite a large amount of their brain because mice basically sense everything through smell. They're really
Uh,
good at that.
yep.
But
Makes sense. They're scared little sniffing rodents.
yeah, they smell better than they see, so you better have a shower if you're gonna like be working with them, or not actually, because it's weird. Because mice are actually freaked out when you don't smell like mouse. So one of the things you
Hmm.
do when you handle them is you rub your hands in mouse poop and be like, "Hey, I'm one of yous," and that really chills them out a bit. Um, uh, so yeah, they look at so this basically you see like various other markers. So they look at Neuropilin-1, how it characterizes to other neuron markers. So
Mhm.
it's usually found on. So the thing that we haven't talked about is how neuropilin is usually found on neurons, and
Yes.
so here
I
they're
guess from
looking
the
at
name, one might expect such a thing.
yeah. But you never know. I mean, there's always a
Yeah.
just another kinase situation where
Absolutely.
Sonic Hedgehog or like sometimes people it could have been named by a fly researcher and suddenly you'd no idea what it does. Um, um,
Yeah, so here they're showing us in the tissues of this mouse olfactory epithelium,
hmm,
which is I guess it's considered neuronal tissue. Oh, I see. No, it has neurons in it, right? Because
yes.
like those neurons feed back into those parts of the brain that sense smell.
Yeah, so this is like in the ethmotobenal. So these would be like in the humans would be quite far inside where the smells are like kind of humidified and then you can actually get the sensing organs in there that
Yeah,
can
yeah.
sense and like the brain and this area in the mouse are very close together. So you can actually see nerves go in between the brain. I mean, when I was working with
Getting
like strep,
swabbed,
we
right? When we get swabbed for SARS-CoV-2, we're getting
yeah,
swabbed all the way back there.
yeah, all the way back there. Um,
And so this is that section of the mouse, and I guess they're using a marker to help us, to identify the cell. TUJ1, right? This is some
So
neuronal cell marker.
yeah, class 3 beta tubulin that is, where it's a you don't really it doesn't, that's not important what it is. I'm
Yeah,
just showing
not
off.
important what it is, just that research knows that it's in the neurons.
Yeah,
And when you correlate that with the NSP RNA, you get correlation.
yeah,
So let's take that as a model then, right?
yeah,
Like, so we've increased our complexity. We went from cells, right, expressing a bunch of different things. Now we're going to take mouse neural epithelial cells, or sorry,
yep,
yeah, and we're going to instill in the mouse's nostrils our various potential binding compounds
yep.
and see what happens when we sack those mice.
So yeah, they use the silver nanoparticles of the SARS-derived CNR peptides, and so this is in Figure 3DE, where they add it to they see how it correlates to neuronal markers. So they use uh, NUN and NUN and aquaporin 4.
Yep. As just looking, I think aquaporin 4, you're going to get the boundaries between cells,
Yeah,
and then I guess the noon, you're going to get the neural cells. Is that what the marker is for?
I think so. I've heard of aquaporin 4. NUN I didn't really hear
Right.
about before, but um,
But the takeaway is that we're seeing positive these cells. We're seeing positive for the nanoparticles, so they're
yeah.
getting in to
So
this.
yeah,
To
the
this.
mouse olfactory epithelium, and now we're going to be looking at the cerebral cortex. So seeing like what happens in does it go all the way into the mouse brain? And
Yeah,
apparently it does.
yeah. These nanoparticles do at
Yeah, the
least.
nanoparticles, which are like much smaller than the actual like SARS-CoV-2 virus, so we don't necessarily know
Yeah,
that it travel in the
yeah.
same way. But
Hmm.
it is an interesting demonstration.
But it is
Um,
possible for something to get there if it can bind, right? Because they're using that CN rule peptide.
yeah,
So, yeah, it's possible to get there if it can bind NRP.
yeah. So that brings us to figure S5, I think, where we're looking at
Just basic. Oh, yeah, I was gonna say they're just using other and our C and R peptides.
yes. Um, So this one looks like there's an association with the cortex, which might be something interesting, hopefully. There's also, well, I mean, uh,
That's all really.
yeah, that's
They
all.
don't. They looked in the corpus callosum. They couldn't find too much.
Yeah, but yeah, I guess these have just like shown as a proof of concept that this cleaved SARS-CoV-2 spike can theoretically travel in this direction. But we haven't
Yep.
tested.
Absolutely, and showing places that it doesn't go as well, right? That's
Yeah,
this is the it's a supplemental, right?
yeah, and that's as important because it'll save you a lot of time when you're actually dissecting the real life SARS virus
Yeah,
because then you won't waste time. You can focus in on the areas that you know can actually have it infected.
yeah. So interesting. I mean, they never do it with the actual pseudotype or anything, right? Like they don't do the pseudotype virus in this case.
No, they don't. I mean, yeah, I don't know
They
what
left
to.
us hanging. They left us. I feel like they left us
Yeah, I
hanging.
feel like
In this line of logic, at least, right? They're gonna go. There's a few more figures left, right? So they're gonna go down some more logic. But in the line of logic of like, can SARS-CoV-2 get into right the human cells? I feel like they're missing some experiments at this end of
yeah.
logic.
Because there are like lots of obstacles that would probably like hamper SARS-CoV-2 that maybe not for the silver nanoparticles,
Right,
and
right,
but
right.
yeah, but I think
But
that
let's keep going.
yeah,
Like
let's
let's
keep going.
because what is attractive about this story, right? Like
Hmm.
why is this in science? I actually think, right? Yes, it is a receptor, but it fits with this with the symptoms that a symptom that we've seen,
Hmm.
right? Like, and that symptom was is the lack of smell.
Yeah.
And so like, and we've seen it before in like a guinea pig model. I think they showed that like it was attaching to the neuronal epithelial cells there, right?
Hmm.
And so it's nice, and they're going to keep going down the path to try to connect it to humans, even though this is where they sort of end the story of their molecules binding in animal models.
Yeah. So in figure S6, they do single cell transcriptomics,
Well, they
where
didn't do it. They're reanalyzing
oh
single
yeah,
cell
that's true.
transcriptomics. Yeah.
Yeah,
So right. So this is like a technique that's been very cool. That's like emerged in from the omics movement. Sorry, I like always like smack my head because like came from an experimental background, so it was always like those people over there that
yeah,
were doing like broad everything. But what essentially happens here is that they found in this case a cell line, right? A cell line, and they sequence all the different cells one by one,
mm,
which is pretty crazy because biology is messy and not. Uh, our previous understanding of biology has been to grind up organs, right, which averages everything together.
yeah.
But these days, you can just you can flow cytometry essentially, right, to get single cells and then sequence each one and see what's going on. Um,
Yeah, they take data from three different papers. So they've got one where they look at single cell longitudinal analysis of SARS-CoV-2 infection, others where they look at just bronchoalveolar immune cells in patients with COVID-19, construction of there's another one where they look at the construction of the human cell landscape at single cell level. So,
Yeah, exactly.
and
Yeah,
they,
so three data sets and that have been made already, right?
yeah,
People have infected these cells with SARS-CoV-2 in this case, right, and run this analysis. And then in the other one, they're already infected, right? It's lavage fluid.
yeah,
And then in the last one, it's like organ level data, which is a little bit different from this. So maybe
mm,
we'll hold off on speaking about that now. But yeah, so they do this analysis where they try to look at the expression levels, right, of the cells that are infected by SARS-CoV-2, right? So I guess it's nice because they are actually going for infection now.
yeah,
It's a cell line culture here, right? Human bronchial and epithelial cells.
yes,
Um, but they see that of the cells that get infected by SARS-CoV-2, they color them in for us,
yeah.
right? Using yellow in this case. And of those cells, you can see that they are expressing NRP1.
Yeah. So this is a weird analysis that I think maybe we should talk about a little bit because this is
Yeah, absolutely. Let's
this
dive
is
in a little.
where data sciences like intercepts with us a lot because I mean we've heard of because growing up we heard about PCA, which is principal components analysis, where you take this complicated set of data that has many like various dimensions and you flatten it out so that you can
Yeah, yeah.
have
Because
it.
what are all the dimensions, right? Because this is the era of big data, right? As I said, they take every cell in the culture, right? So you have a dish full of cells. You dissociate them so they're single cells, and then each cell you are doing deep sequencing on to find out what genes are being expressed, right? What RNA is in that cell at the moment, right? And that means in terms of dimensionality, right? You have one point of data, and for each point of data, each cell, you have the entire list of genes, right, of RNA that's being made in that cell, right? And so, how does one graph that object? You have to reduce the dimensionality, right? You can't have a graph where every axis is yes or no on a different gene. I mean, you can.
Mm,
That's essentially what A is,
mm,
right? For a single cell, right? You could look at for every dot on this graph, you could look at a graph that looked like A, but that is a lot of data to look
yeah,
at. So instead, they use these methods of grouping things, basically, right? Dimensionality reduction is trying to group it, but without pre-assigning the groups,
mm,
right? It has to be agnostic to the actual content of the data, right? It can only just show us the groups based on the structure of all of the data itself.
yeah.
So I don't know. Did you want to explain
I
what this
mean,
UMAP is?
all I know about it is called Uniform Manifold Approximation and Projection Visualization. So
Mm.
it's so I know that dimensionality reduction is a big thing in the data analysis community because I mean everybody has data. I've got data right now. I'm on YouTube and I'm on OBS, and they're all
Yep,
collecting stuff about me, and then they pull me down to one data point so that I can group into certain areas. In the same way, these cells have been
absolutely.
grouped into certain areas based on. So it isn't just like one gene. It's almost like all of the set of behaviors have been encoded into one area part of it. So
Yeah, absolutely. Yeah, that's a good way of describing it. I One important thing to remember with this particular form of dimensionality reduction is that the distance between points doesn't matter.
yeah.
Which is kind of crazy to think of a graph because I think we all grow up in Cartesian space,
Mm,
right? Where graphs are move three across and three up, right? That is the data point. This is not in that sort of space, right? It is sitting on paper. You could draw lines and look at distance, but the distance between each point doesn't matter that much. What matters is the groupings of points that occur,
mm,
which is very strange, right? That's like maybe counterintuitive for people reading these
mm.
things, right? You might be like, oh, but this group is closer to this group. I mean, yes, in general, but nothing from that distance means anything. It just means there's distinctions. Distance in this graph means distinction, but it can't really tell you anything more than that. So like the sizes of the groups don't really matter, right? Also, like because the more data you have, it'll just put more data points in those groupings. Um, yeah, it's a weird space to look at, but I like to think about it as white space matters a lot,
Mm.
right? If you can see white space isolating groups, then that gives you a good idea of the clustering,
Yeah.
right? Like so that's how I try to read these. Is basically I do see groupings,
Mm.
right? Your eye cannot deny there are groupings,
Yeah,
right?
I see groupings.
And those groupings are defined by the white space around them.
Yeah.
And then if you can see the little lobes, right? Now you know, okay, so there are groups of cells that get infected,
Mm.
and you can follow up, right? You can say, what is that?
Yeah.
What's going on there? Yeah, I don't know. Like, what is D exactly in this graph? Let's read the figure legend. I feel like that's gonna give us. We could talk slightly more about this. Heat map showed the average expression of NRP1, furin, TMPR, SS1A, and ACE2 in the ciliated cells of mock-infected and SARS-CoV-2 infected. Okay, yeah. So it's
Mm.
really just an overall heat map of all of this. In this case, all this dimensionality reduction isn't helping. Right, we're not learning that much actually. The
Yeah.
dimensionality reduction here.
It's
Um, yeah, they could have just shown us the heat maps. I think. Um,
yeah, but I feel like if they've done all this work, they want to show us
yeah,
something. They
yeah,
because
yeah.
but yeah, I can sort of understand the need, but and also it's
Yeah,
a
it's gonna
lot.
set us up for talking about these graphs later, right?
Yeah.
Because dimensionality reduction matters a bit more in the other ones. In this one, um, not really. I would say.
Yeah. So this one is another one where they look at NLP1, NLP2, and ACE2, um, and
Yeah,
seeing how
yeah,
they
seven. We're looking at seven.
yes, S7.
Yeah, yeah. So the different data set, different cells now.
Yeah.
Bronchial lavage fluid.
Yeah.
And so, I mean, to me, this is a great. I love these,
Mm,
right? Because this is like from infected patients.
yes.
These are human cells that they're reanalyzing in light of NRP1.
Yeah.
And again, they don't have to do. They don't have to do the dimensionality reduction. That's not the point of what they're showing us. They're just showing us that SARS-CoV-2 positive cells have NRP1. Yeah.
Yeah.
Thank you for the,
So
but not
yeah.
necessary.
I mean, they kind of show that it's enriched. They take care to not say it's like elevated. They because it could be that the SARS-CoV-2 could potentially be upregulating NLP1 production, or it could be that cells that express lots of NLP1 have a lot of SARS-CoV-2 because they're more liable to be infected. But it kind of establishes that correlation.
Yep.
Uh,
That's definitely what the work that they're doing, right? They're correlating, right, the expression of SARS-CoV-2, or yeah, so the expression of SARS-CoV-2. They're correlating the infection of SARS-CoV-2, right, with the expression of these Neuropilin-1.
yeah.
uh, okay, here's something where I think the dimensionality reduction means a little bit more, right? Because now they're just trying to say what cell types are actually being infected potentially, right?
Yeah.
Uh, they're
So
go. I do think that they're doing this analysis sort of like at risk, right? Like they're going out on a branch on a limb,
mm,
right? Because like they're assuming that we've bought the principle, right? That NRP1 um, is physiologically relevant in
yeah.
infection. For yeah, and from that now they'll ask very tentatively, right? What cell types could those be? If you buy the premise, right, that it is infecting these cells.
So in this case, they use a. So I've tried to label the different groups to check because they look at different like they look at a sample of like lung cells and they break it down into different groups by color. The problem is they use
Yep.
multiple colors for certain groups, and it's very by eye it was very difficult for me to tell which ones are which. So some of these labelings might not be perfect, but
Yeah. Well, I think the reason why, right? Like, it's nice to see this. It's like the reason why they did the color spectrum, right, is because how they chose the colors were just from like definitions of cells
mm,
that people have found in the literature, right?
yeah,
They define cell types as expressing this suite of things. So they put similar cell types together to make it easier for us to see in general what's going on. Are we looking at figure S8, right?
yes, figure S8. I accidentally copied two, so the second one is the one where I
Second.
put in
Yeah,
labels.
I like the second one. Yeah, the labels are excellent.
So
Um, yeah, so that's what they did. That's A is basically the work that you helped us do is that you took A and you summarized it for us, right? You took similar colors and bringed us together. But you can see like there are things that mix, right? Like myeloid cells are in like almost a similar cluster,
mm,
right, as um, endothelial cells. Oh, they're using the outdated. Oh, you updated it.
yeah,
Beautiful.
I
You
updated
updated the
club
wording.
cells.
Club cells. Yeah,
Yeah,
wonderful.
because I saw that, so I was like, wait a minute, isn't it there a boo boo? I don't like. Do we want to really like lionize this person's work? And like, no, we
Yeah,
don't.
yeah. But like that's the thing. Like, I mean, sorry, this is gonna be a little tangent. We're already a
Go
bit
for it.
over time, but like the tangent is algorithms, right? The people who do this stuff, they're just running the analysis. This
Yeah.
database doesn't care about like our most recent understanding of the world and science, and so like someone needs to go in and edit the annotation algorithm that they used here and change that Clara cells to club cells because that's what we're calling them now.
Yeah. So I've put up this like kind of atlas of like what the cell types are next to the NRP1, so you can sort of see like the red is where it's most expressed in that NRP1, and orange is kind of medium term, and you can see that there is quite a lot expressed on the endothelial cells and
Yep.
epithelial cell like kind of regions, which are quite important for SARS-CoV-2 infection.
Absolutely, absolutely. Yeah, like they're really trying to bolster their idea, right, of this by giving us these correlations, a whole cloud of correlations.
Yeah.
And that's why I think that these things make the supplementary figures but not the main figures
Yeah.
is because like it is just a lot of correlation. And we don't have that hard experimental evidence, which is what the publications are for. But nevertheless, this is very important work because it is helping us build that hypothesis, right?
Mm.
NRP1 could be this thing that drives the symptoms, the neurological symptoms.
Yeah, I mean, another version of this paper, I can imagine this being the first figure that introduces us to the hypothesis of could NLP1 be significant for SARS-CoV-2
Yes,
infection?
yes, yes, absolutely, absolutely, absolutely. And it's great because it takes work to make these, and I'm
Mm.
glad that they did this, and yeah, helping us see it as well.
Yeah, I
And so
mean,
they do the same thing. Maybe just
so
plow onto S9.
yeah, so S9, I've done it a bit differently because I just literally was running out of time, so I just put arrows and circles.
Beautiful, but same idea, right? Like they've done this clustering this time of the olfactory neural epithelium, right?
Mm.
To show us that, okay, here are the types of cells that have a lot of NRP1 to drive a hypothesis, right? That they haven't fully shown us yet, right?
Yeah.
That they're just like building that that it could be infecting these cells, and that's why we get certain
Yeah.
neurological symptoms.
I mean, one thing I want to point out is that there is like one area that says it could be infecting vascular smooth muscle or unidentified. They're both very similar shades of pink, so it could
Yes,
be.
yes.
But yeah, but again, that builds up that hypothesis of
Mhm. Okay, and what I like and how they're wrapping up this paper, right?
mm,
Right, I think that their logic kind of broke down, but they're really trying to drive towards the human aspects of this,
mm,
and so they go into actual or human biopsies, um, and they start staining. So
mm,
in actual human cells, so this is in some ways this is their experimental evidence to confirm their correlations that they found in Figure S8 and
yeah,
or S9. Just like if they actually take biopsies of this epithelial, do we see um, expression of NRP1? Yes, they do.
yep, they do. In fact,
They also see expression of these two.
yeah, Ace too. Because I think like they see like quite a lot of expression of NRP1 in comparison to ACE2.
Yeah, absolutely. A lot more NRP1 than ACE2.
Yeah,
And then their sort of CUDA, their prize their prize gem in the relevance data, right, is Figure 4 and Table S1, where they get a hold of patients who are actually infected with COVID-19 um, uh, and pass away. I think, right?
yeah,
These are autopsies. Um, and they get a biopsy of their olfactory epithelium and they stain for both NRP1 and the spike protein.
yeah. This is an interesting one because the NRP is in purple, S protein is in brown, but
Hmm.
like everything is in purple. Weirdly, like I mean,
A lot of purple.
yeah, and
There's a lot of purple.
yeah,
If you look back, you can see, right? Like if you look back at S10, there's tons of brown,
yeah, that's right.
right? This villain's everywhere. Um, so yeah, again, the story is not complete here, right? They're
Yeah,
sure of showing us a lot of different things to drive us towards this hypothesis. I think it's a very compelling hypothesis,
yeah,
right? They've done a lot of very good correlation with us. Um, and I guess just let's look at S11 before we try to wrap up this paper altogether.
yeah,
They threw us another little just fragment of of suggestive information where they say, oh well, like let's think of this like differentiation marker, I guess is what they're looking at,
hmm
OLG2, and showing that oh yeah, like this correlates well with the the COVID-19
yeah. Olig
in
two is for neuronal cells. I think it's yet another one for neuronal cells.
neuronal
Um,
cells. Yeah, yeah. They really want to drive us to this point, right? That that we have seen SARS-CoV-2 get into neuronal cells, right? In human samples, right?
yeah,
These neuronal cells have NRP1, right? And then through an incredibly rigorous set of experiments, they've showed us that the spike protein of SARS-CoV-2 can bind NRP1,
mm,
right? And in fact increases the ability for pseudotype particles to enter those cells.
yeah. I think they've painted a good picture, but like towards the end, like in human things, they almost overplay their hand a little bit, like where they're
Totally
yeah.
overplayed their hands so they can get into science. Sorry, I like always say that.
Yeah, I mean, I feel like having two papers on the same thing at the once kind of helped the situation. So
Yes, yes.
I mean,
It helps the situation in that we know that there is this other receptor. I still really want to know, like, what is its significance in some sort of disease process? They don't have the phenotype to
yeah,
me. To me, they don't have the the testable phenotype that they can bring into an animal model and then tell us, like, what is the effect in this sort of infection.
yeah. It felt like they stopped just short of that interesting like idea because that's what
Yeah,
because once they go
yeah.
there, you can think about treatments and other sort of situations. But right now, it's like a cell culture model that correlates to an effect that happens in humans. But again, we don't know that necessarily it will make that leap because I mean,
Yeah.
SARS-CoV-2, yeah, it's enriched in epithelial cells, but do you know what else is enriched? But that's like the main contact for SARS-CoV-2. So
Exactly. That's where SARS-CoV-2 is everywhere. It's always like flowing around those epithelial cells, or those and yeah, epithelial cells.
yeah,
That's
and
the outside world. That's the epithelial cells are the outside world.
and this isn't the only like kind of theory for why like SARS-CoV-2 goes into neuronal cells. I think there's other like ideas out there because I remember we explored one in that hamster paper where that got someone infected. But it's a compelling hypothesis. I think they've really
Yeah, yeah. I think the other thing that I like about it is because they do some of this, because they well characterized the biochemical interaction, right, between the spike protein and NRP1. It actually, to me, it paints the single cell sequencing data of figure S9 and S8 in a different light when creating your own hypotheses about, right, like for other scientists to sort of create their own hypotheses of what they think could be happening in infection. Because as we saw in the introduction, there's so many different symptoms that have been reported as a result of these of SARS-CoV-2 infection,
Hmm.
and those symptoms can all have their own mechanism of occurrence. And this information helps to start build maybe a mechanistic
Hmm.
hypothesis as to why those symptoms occur. Particularly, I'm actually interested in this idea of all this NRP1 on macrophages,
Yeah.
because we know like the immune system plays it a part. And earlier we had a paper, right, that was talking about inflammatory signaling pathways in macrophages as it relates to the SARS-CoV-2 infection. And I would like to see papers come out, right, that follow up on that sort of work, that start tying in this NRP1 story into the immune system
I mean,
response.
yeah. I mean, it'd be very hard to pick out because if you've got someone who's infected with SARS-CoV-2 and their macrophages don't have SARS-CoV-2, then you've got a problem. Then you understand what the problem was. So I mean,
Exactly.
so
Exactly.
it's hard to
Hmm.
like pick out NRP1's role, NRP's role in that
Yep.
situation
Yep.
because you're like,
Absolutely.
okay, yeah, macrophages should have SARS-CoV-2. So you can't anything on there. Anything on a macrophage surface could correlate to whatever virus you want. I mean, so
Absolutely. Absolutely. Yeah, so that is an incredibly challenging area to work in, but
yeah,
to me, like that's what excites me about this paper
yeah,
is that its implications on cell signaling pathways potentially, right, that we just are aren't articulating right now.
yeah. I mean, it opens up quite a lot of like ideas because I think the another thing that's like kind of usually thought of is that once like SARS-CoV-2 is like cleaved, the fusion peptide, the S2 part, is in the
Yeah.
membrane, but the S1 kind of floats away and doesn't do anything. But
Yes.
that's the
Yes.
part that has the C end R like peptide. So,
Yes. So here's a use for it.
well, I'm just trying to understand what that use would be because it'd be floating around and then it's like hit Neuropilin-1, but at this point it's not attached to SARS or anything. It's just floating around on its own. So, is it doing anything? Or I mean, this
Oh,
is something that
no, no,
it,
no. Actually, no. That to me, in the viral life cycle, this makes sense, right?
right?
Because it's efficiency, right? S2 is the fusion peptide.
Mm-hmm.
Is that what I'm understanding? I
Yeah.
actually don't have that clear in my mind.
Oh
Okay,
yeah.
S2 is the fusion peptide. So S1 is basically just dead weight
Yeah.
unless evolution drives it towards a useful thing, right? And in this case, it's a useful thing because it can act as a co-receptor,
Mm.
right? You can bring activated spike in close proximity to Neuropilin-1 or whatever, right? In the hopes that it would then increase you could then get binding to the ACE2 receptor and then do fusion,
Yeah, I think,
right?
and another thing is like since SARS-CoV-2 is in a triplet, it's like
Yep.
in a trimer. So
Yep.
one of those things gets like cleaved, then it might not float away. I mean,
Hmm.
we don't really have much visualization on that specific process. Because if it's like a
Right.
stepwise cleaving, then yeah, it could still act as like a binding process. But
Hmm.
if it's all
Yeah,
cleaved,
that's a great point. We don't know when
yeah,
in an infection does the S1 S2 site get cleaved by furin by TMPRSS, right?
yeah.
When
Do
when does it happen?
I mean, do all the things need to be cleaved at once in order for the fusion proteins to work, or can only one need to be cleaved and then the fusion? So
Sure.
it's some
Oh, yeah, that's also very. You know what? We may have a paper to help describe that a little bit more because I think that they've crystallized post and pre-fusion complexes.
right.
So they
Ah,
may look at, yeah, they may look at whether or not it's required for all three fusion peptides to be open or just one. Anyways, that's just
yeah,
add to the long list of papers that we could dive into, right? Like that may be in some of the literature that we've already looked at.
yeah.
We just haven't dove into it yet to know the answer.
Yeah, I mean that's the great thing about papers like this is like they do raise. I mean, the great and also kind of frustrating thing as well if you're a layperson, where they raise more
Yes.
questions at the end of them.
Yes, but that, that is what characterizes, I think, in my mind, like the best basic science, right? Is to sort of give us a new nugget of information that we can talk about very concretely, but then open it up into the whole wide world of hypotheses
Yeah,
and say, figure it out yourself.
yeah. This is the kind of science you can put in a grant application. And go, hey, there's still questions open up, and it got them a Nature paper. It might get us a Nature paper.
Yeah, absolutely. And yeah, and I think in terms of understanding things about viral biology, you know what? Almost like the types of questions that we've been talking about, be like that are facilitated by talking about this. There's amazing principles of viral evolution and biology at play here, right?
Mm,
Like this is going to be SARS-CoV-2 is definitely going to get into the virology textbooks,
mm.
right? Has some really amazing stories of how viruses get shit done.
Yeah,
Right? Because like even the story of like one peptide is a fusion peptide, the other peptide is potentially a co-receptor ligand, right?
yeah,
Amazing.
yeah. That's. I mean, I feel like all this like kind of magnifying glass that's been put over SARS-CoV-2 is like it's going to still be relevant for other viruses that are very similar and be interesting for also for the way we analyze things. I mean, the 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 fact this paper tried to bring in a whole bunch of data analysis that was almost like completely like new to me was
Yeah, yeah. It also shows you the way science has gone, right?
yeah,
Uh, Good science and very successful science these days is a combination, right, of experimental evidence as well as reanalysis of large data sets that exist. And being able to build a hypothesis from the combination of those two is what will make a scientist successful these days. But you can also do all the little bits and just communicate what you've done.
yeah,
All right, open science advocates here, right? Like
yeah.
any one of these little things could have been published into the void, right? And then teams of people could have absorbed those things, contacted each other, and begun to work on a project.
Oh yeah, and all the for specifically the UMAP data, all that data has been made like available, right? So if you're a data scientist and you feel like you want to do something, maybe you could analyze that and find
Yeah,
spurious content.
find some scientists with questions, right, and
Yeah,
help them out, or find groups. I'm always talking about citizen science. Find your local citizen science group, right? Your DIY biolab, right?
yeah,
Find people there that might be willing to collaborate, or at least link you to collaborators, and like you could find yourself doing some of this work.
yeah. Like if you're in New York, maybe Biotech Without Borders.
Absolutely.
Slight plug there.
Type plug. um, yeah, but I think that's great. Do you have anything else to say you want to say
No,
about?
I think I've said my piece on that paper, so I think it's been really great having these conversations. Really great to geek out with you on these. Uh,
Mm.
so yeah,
Absolutely. If people listening along are interested in suggesting papers that we should comment, or have listened to our rambling and find that there are errors and inconsistencies in the way that we talk, please tweet us, comment us in the below. We'll do our best to rectify any of those problems
oh
because we believe that science is best done in the open, and we are just part of the peer review process here with all you guys. So yeah, reach out.
yeah, and also like, comment, subscribe, hit that bell. Um,
Yes.
yeah,
See you guys next week where we'll cover microbiology news and pick our paper for the following week after that.
yeah. I've been Fazalam.
And I'm Danny Chan.
We'll catch you next week. Bye.